Hello everyone, it's Dr. Wolfe here. Sorry I can't be in class today. I really wish I could be, but I wanted to create some videos to give you guys some guidance on what you're going to be working on today. So just a couple things. Today, just a little bit of guided practice with videos on cladograms, and then there's just a bunch of other practice with electrophoresis cladograms as well. A couple things. Formative is going to be on Monday on cladograms and electrophoresis. You'll notice it's just on SCP-8. There was a little bit of switch there with the team. There was some mislabeling and everything. Nothing's really changed in terms of what we're learning, but it's just going to be SCP-8, which kind of makes sense. We're not really necessarily doing a whole lot of mathematical calculations. Tuesday is going to be our summative. Same exact thing, SCP-8, electrophoresis, and cladograms. So one other thing I wanted to do is show you guys visually the tracking sheet here. So I know nothing's changed in terms of what I've communicated to you all. It's 7 scores per category, right? But at least this gives you a little bit of a visual. This is the one that I'm going to be grading this weekend that we took earlier this week on replication and cell cycle. And then this is the one that's going to be on the Tuesday right before Thanksgiving break. When we get back from Thanksgiving break, we're going to be diving into photosynthesis with all of these skills. And then you will have one other summative right here, cumulatively on all the skills that we've been learning for SCP-5 mathematical reasoning. Okay, so yesterday you guys created cladograms, all right, and these were essentially the two main cladograms that I saw at the whiteboards that you guys made, right, where really the difference is whether the cuttlefish and octopus is more closely related to humans here or more closely related to starfish, right? And the reasoning, the evidence, is the known characteristics. Lion and human, their common ancestor must have shared fur because these ones don't have it. The cuttlefish has this self-control, this intelligence, and we know that humans, lions, and are pretty intelligent too. Do the lions, how do they do on the self-control test? We don't have evidence for it right now, but we at least know humans and cuttlefish can do that, right? And we know that lions share fur. All right, starfish, and all these organisms, they are multicellular tubes. Their common ancestor was a multicellular tube. Now over here, similar thing, right? Fur is a common trait of the common ancestor, multicellular tube. The reason I left this blank here is just different groups had different answers. So our goal today is to figure out which one of these cladograms is correct. Now, how do we determine that, right? Our evidence here is known characteristics of animals, but that's kind of it's difficult as you guys were talking about it with your groups, right? Do we put this common trait, is that fur, or is it the fact that they're vertebrates, you know, that have a backbone? Well, both are true and both technically would work. So there's a lot of different pieces of evidence you could look at. What we want to do is confirm it with molecular and DNA evidence, all right? So how we could do this is this concept called the molecular clock, and I'm going to introduce it with this specific example. Humans and chimpanzees, they both have this hemoglobin protein, all right, that carries oxygen through their bloodstream. Same protein, does the same job. However, if you look at the DNA sequence for hemoglobin in chimpanzees and the sequence of hemoglobin for humans, there's some differences. So the question is, why? It still does the same thing, right? Well, let's say humans and chimpanzees share a common ancestor. It's 6 million years ago. So ignore this 25 million years ago. That's kind of copy and paste from another source. That would be 3 million years ago, or 3 million years ago here. This would be present day. So 6 million years ago, common ancestor. Humans and chimpanzees don't exist. Here's a section of that hemoglobin DNA for our purposes. And then those populations split, right? Now, let's say like in 3 million-ish years' time, there's a mutation that accumulates in the human population, all right? And then like all humans kind of eventually have this mutation, and another mutation happens in the chimpanzee population in another region and kind of gets passed down and accumulates in that population, right, where it's spread there. And then you can imagine over time, mutations and mutations build up. Now you might wonder, well, doesn't a mutation affect a protein? Yes, and no. Don't worry about knowing this chart for this unit. This is a little bit of foreshadowing for next unit, but essentially what it's showing is that you could have this, here's an amino acid, and you could have different codes for the exact same amino acid. So let's say my RNA is GCG, which came from the DNA, gives me alanine. I could have GCA, all right, GCC, GCU, and I still get the same amino acid. So that's why you could have mutations and no change to the protein, all right? So All right, this brings us to the molecular clock. It's this concept in biology, all right, that mutations occur randomly in DNA over time, and in some regions, like non-coding regions or neutral regions, those were the regions that it's not going to change the amino acid, these mutations do not affect survival and tend to accumulate at a steady rate. So this predictable accumulation acts like a clock to measure time, all right? The clock is calibrated using known evolutionary events such as the fossil record. So for example, if two species, like we know from the fossil record, boom, their common ancestors 10 million years ago, we look at their genetic differences, there's 5, well then we could estimate that there's 1 mutation per 2 million years. So same thing over here, let's say we have 25 differences in this area of the DNA, we can predict that they shared a common ancestor 30 million years ago. Now, this molecular clock is not the exact same for every single gene. It depends on it for different coding regions, right? So it's not this like universal rate, but if we dive more into like specific regions, we could figure out what that rate is and apply it to kind of figure out when species diverged. Now, we're not going to be using the molecular clock to say like this is when they shared a common ancestor with cladograms right here. Yes, this shows time that as we go further down, right, that we're going further back in time, but with cladograms, at least the ones that we're going to be making, right, it's not to scale, right? It's not saying that like this would cuttlefish and octopus, their common ancestor was this much more time, like three times back was the cuttlefish and octopus compared to the lion and the human. All right, it's just kind of general what we're making. So what we're going to be looking at is the differences in the DNA. All right, so what I want you guys to do is on your sheets, you have the same exact table. I want you to make a cladogram based on this DNA evidence, all right, and see if it matches with the one that you guys made above. Again, how we could start to make this is looking at the differences in DNA between all these different species, all right? I would maybe start with humans, seeing like what's most close with humans, and then kind of build from there. All right, good luck.